KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video we are discussing in continuation of meiosis. So let us start with the anaphase 1. So what is information we are giving now? In previous video we have learned the information in meiosis 1. 4 stages we are observing now. So what are those? Prophase 1, metaphase 1, anaphase 1 and telophase 1, isn't it? Now there, in previous video we have discussed the prophase 1 and metaphase 1. In this video we will discuss regarding anaphase 1, anaphase 1 and telophase 1. After completion of these two phases we will enter into the meiosis 2. In meiosis 2 we will discuss about the prophase 2, metaphase 2, anaphase 2, telophase 2. Easy only, no? Meiosis 1 there 4 stages, as well as in meiosis 2 also 4 stages. Avalada, adi the meiosis. Okay, so here the somewhat like a diagrammatical explanation also I am going to give you sorry, by the end of the video. Now in meiosis 1, anaphase 1 there, the homologous chromosomes separate. Main important points enna suppose this much of information in absence of teachers, suppose if you are facing any kind of burdens to understand, so simple clues I will give you, so just try to save in your mind. Prophase comes, prophase nalli what is happening, no? Chromosomal material will be there, no? Chromatin material, that particular chromatin material condensed and converted into chromosome. That information mattum nalla save pannikonga. Where? In prophase. Next one, metaphase. So in metaphase, so what are the chromosomes are there in the cell? No, so all those chromosomes will come and sit at the or align at the or arrange at the equatorial plate. Avalada, adi metaphase. Next one, anaphase. So due to the bipolar spindle fibers or due to the microtubules activity, so chromosomes start to move towards the opposite directions. Adi anaphase. Next one, telophase. Telophase opposite to the prophase. Means what are the characteristics we have seen in the prophase? No, all those characters we will observe in opposite conditions in case of telophase. So that's why here once you see in anaphase 1 the homologous chromosomes separate. Now only we see, no, in anaphase 1 chromosomes get separated. Okay, and while the sister chromatids remain associated at their centromeres. My dear students, here what is the main important point we have to remember? No, so in anaphase 1 actually if you take mitosis, no, there the chromosomal number or what is a chromosome is there, no, that particular chromosome that will be splitted along with the centromere in case of anaphase of mitosis. But here in anaphase 1, what is the information we are observing? Chromosome is not going to be divided. Chromosome so contains of 2 chromatids, no. 2 sister chromatids are there. That one is not going to be divided. That is, it remains associated with their centromeres. Okay, so thereby here what is the information we are understanding? In anaphase 1 the homologous chromosomes separate and while the sister chromatids remain associated at their centromeres. Next one, telophase 1 point of view. So what are the main important points? Means the nuclear membrane the nuclear membrane and the nucleolus reappears. So already we learned no. So telophase means which is definitely opposite to the prophase. In a prophase, so we have observed no disappearance of what we have seen nuclear membrane and nucleolus we seen. Now here what is the point we are observing in telophase? One, so nuclear membrane and nucleolus reappears. Reappear and cytokinesis follows, and this is known as a dyad of cells. So means after completion of telophase 1, so these two phenomena, one is nuclear membrane and nucleolus reappearance and followed by the cytokinesis. Definitely cytokinesis, after completion of cytokinesis, one part of meiosis will be completed, that is meiosis 1. At the time how many number of cells of formation takes place? 2 cells. That's why here this one we will be taking as dyad of cells, DYAD, dyad of cells. Next one, although in many cases the chromosomes do not undergo some dispersion and they do not reach their extremely extended state of the interphase nucleus. Means here, although in many cases the chromosomes do undergo some dispersion, so definitely they undergo some somewhat dispersion and they do not reach the extremely extended state. So they do not reach the extremely extended stage of the interphase of a nucleus or interphase nucleus. Okay, next one, the stage between the stage between two meiotic divisions is known as intrakinesis. So what is the stage? So here we are studying meiosis 1 is there, after meiosis 2 is there, we are studying so there after completion of meiosis 1, so before start to division for meiosis 2 or before entering into the meiosis 2, somewhat gap will be there. That particular gap we will be considering as interkinesis. Don't forget NEET examination point of view important. Intrakinesis is nothing but which is situated, or the gap which is situated between the meiosis 1 and meiosis 2. And generally it is short-lived. Generally it is short-lived. What is the meaning of this one? No, after completion of meiosis 1, konja gap will be there. Again soon meiosis 2 will be started. That's why here the interkinesis lifespan is, or the generally it is short-lived. Next one, there is no replication of DNA during interkinesis. So this particular point also you have to save in your mind. So there is no replication of DNA. So in this particular case, sorry, gap is there, no. So that particular gap again chance is there sir, for replication of DNA means no chance at all. So that's why here one particular point we have seen. So already we have seen what is the, so before meiotic division, so DNA replication process takes place, and in between the meiosis there is no chance to the DNA replication we see, isn't it? So that's why here what is the point we are understanding? No, there is no replication of DNA during interkinesis. Next one, interkinesis is followed by Prophase 2. Means once interkinesis path, interkinesis where it will be present between the meiosis 1 and meiosis 2. Why? Because after completion of meiosis 1, so from the meiocyte, from the parental cell, 2 cells of formation takes place. Those 2 cells we are calling as a dyad of cells. If 2 cells of formation takes place, once when 2 cells are formed from the meiocyte by the process of meiosis 1, konja gap again meiosis 2 start out. So meiosis 2 definitely will be there after the interkinesis. So that is only here we have written. So interkinesis is followed by Prophase 2. Okay, next one followed by Prophase 2, a much simple or the simpler prophase than the Prophase 1. So here this particular Prophase 2, sorry, this Prophase 2 very customer sir, means like a Prophase 1. Likewise here also Prophase 2 also like a leptotin, zygotin, packaging and the mother stages sir, means no stages, nothing. So that's why this particular prophase 2 is very easier than the prophase 1. Why? Because in prophase 1 we are observing, or we have seen 5 different substages, isn't it? Leptotin, zygotin, packaging, diplotin, diakinesis. Likewise, so many different types of steps or somewhat depth information will learn in prophase 1. So such kind of depth information is absent in prophase 2. That's why this particular prophase 2 is a, or it is a much simpler prophase than prophase 1. Easy amma adida. Sorry, right. Now let us start to discuss the meiosis 2. So there in meiosis 2 also again prophase 2 will be there, metaphase 2 will be there, anaphase 2 is there, and one more is telophase 2 also is there. One important point I will give you, try to save in your mind. What is that? No, meiosis 2 is equals to mitosis. Meiosis 2 is equals to mitosis. So thereby, so what is the meaning here we can understand, no? So what are the offsprings? Means here what are the cells involved in a meiotic division? What are the cells involved in meiosis 2? Definitely, so produces or what are the offsprings which are produced from the cells of meiosis 2 definitely maintains the equal or the same number of chromosomes. This is a lengthy sentence I have used, but now once you see there, so thereby I will tell you. So that, but here one point save pannikonga. What is that? No, meiosis 2 is also known as mitosis, equals to that mitosis. Okay, now let us start to discuss prophase 2. Already I told you, no. So what are the main important characters? Say here we can see in prophase 2, metaphase 2, anaphase 2, in telophase 2. No, what are the main important characters? If you take prophase 2 there, thread-like chromatin material condensed and converted into chromosomes. Apro prophase 2 point of view, one more, what is the point? Definitely nuclear membrane and nucleolus disappears. ரெண்டு பாயிண்ட் நல்லா சேவ் பண்ணிக்கோங்க. அப்புறம் மெட்டாஃபேஸ் 2 what is the point? So what are the chromosomes are present in the cells, no, will come and sit at the equatorial plate. Equatorial plate மேலே arrange ஆகும், அவ்வளவுதான். Next one, anaphase. So chromosomes, so what are the chromosomes are there, no, in cells, no, all these chromosomes splits. Now here the chromosomes, so splitting process we will observe in anaphase. அப்புறம் telophase write பண்ணும்போது or learn பண்ணும்போது இது remember பண்ணிக்கோங்க. telophase 2 is opposite to the prophase 2. So thereby by the process of decondensation in telophase only, by the process of decondensation, chromosomes converted into thread-like material and reappearance of nucleolus and the nuclear membrane we will observe. Once the telophase 2 also is completed, we will observe the cytokinesis process. That is only, no, இப்போ karyokinesis அப்புறம்தான cytokinesis. So karyokinesis is nothing but presence of 4 different types of stages: prophase, metaphase, anaphase, telophase. Okay, now let us see the information of prophase 2. So this particular information எல்லாருக்குமே easy தான். Once you see there, meiosis 2 is initiated immediately after the cytokinesis, usually before the chromosomes have fully elongated. See there, so definitely, so in meiosis 1, after completion of cytokinesis process and after completion of cytokinesis process, immediately we will observe the initiation of prophase 2. So there, what is the point we can observe? Chromosomes have fully elongated. So very clear structure of prophase 2 or very clear structure of chromosomes we will observe in prophase 2. Next one, in contrast to meiosis 1, so here the differences or the somewhat differences are there in meiosis 1 and meiosis 2. Now here once you see, in contrast to meiosis 1, meiosis 2 resembles as a normal mitosis. So that's why I said meiosis 2 is equals to mitosis. Now here the nuclear membrane disappears. By the end of prophase 2. So definitely at the end of prophase, definitely we will observe the disappearance of nuclear membrane and nucleolus. So here also in prophase 2 or by the end of prophase 2, we will observe the disappearance of nuclear membrane, and one more also here we can take, that is nucleolus. Next one, chromosomes again become compact. You see there, the condensation is there now again. So by the process of condensation, so definitely chromosomes, thread-like chromatin material condense and converted into chromosomes. Simple amma idu ellame. So what are the main important points we are discussing? No, all are most important points that which we have to learn definitely. Next one here. Next one, if you see there, no, metaphase 2. So metaphase 2 point of view, at this stage, so at this stage, the chromosomes align at the equatorial plate or equator. Point why? Because we have learned metaphase point of view, in this particular stage, chromosomes align at the equator or the equatorial plate. Nethi we learned equatorial plate, and the microtubules from opposite poles. So opposite poles of the spindle get attached to the kinetochores of the sister chromatids. So thereby definitely on both sides of, or from the both regions or from the both polar regions, so which one will come? Bipolar spindle fibers comes, or microtubules. Our knowledge point of view, these microtubules will come and binds at the kinetochore region. If kinetochore region bind upon next time what is happening? So when these bipolar spindle fibers, so first of all I will tell you, in a metaphase 2, what is happening? When these chromosomes will come and sit at the equatorial plate, now from the both poles, which one is coming? Bipolar spindle fibers or the microtubules or the spindle fibers comes towards the chromosomes and get attachment with the kinetochores. Once these when these bipolar spindle fibers attach to the kinetochore region, automatically these bipolar spindle fibers helps for the separation of the or splitting of chromosomes. So now here what is the point? In anaphase 2, this particular anaphase 2 begins with the simultaneous splitting of the centromere of the chromosome, allowing them to move towards the opposite poles of the cell. So easy point only. Now here what is happening? Anaphase 2 epo start avana. So during anaphase 2, due to the activity of spindle fibers, what is happening? One particular chromosome that will be splitted. Now, one particular chromosome structure we know very well. So that particular chromosome center is split. Even centromere also split. But my dear students, listen one point carefully. Now anaphase 1 and now we are learning anaphase 2. What is the difference? In anaphase 1, there is no splitting of chromosome. The point. In anaphase 1, there is no splitting of chromosome. Okay, there is no splitting of chromosome and there is no splitting of centromere. Okay, why here? That's why here, for example, here one example I have taken once you see. Here, how many number of chromosomes totally we took here in this particular diagram? 2 and number 4 we took. Now, anaphase 1 we see previously here anaphase 1 we see total number of chromosomes in anaphase 1 2 moving to that side and 2 chromosomes moving to the another side, isn't it? That's why in telophase 1 you see from this cell here we have seen the development of 2 cells. Cells or gametes once you see there. In this particular cells now how many number of chromosomes are there? 2 and here 2. But totally initially how many we have taken? 4. But here we took 4. But here 2 only are there. Here 2 chromosomes and here 2 chromosomes. So thereby in meiosis 1, once you see the diagram. So from here to 1, 2, 3, 4 and 5th diagram also here you see there. Up to here, this entire process here we will be taking under meiosis 1. Why? Because once you see here prophase 1, metaphase 1, anaphase 1 and telophase 1. So up to here, so we will be considering under meiosis 1. Here totally first cell or the diploid cell condition, how many number of chromosomes we took? 4. But after completion of meiosis 1, each cell, how many number of chromosomes are there? Here 2 are there and here the 2 are there. So thereby after completion of meiosis 1, so the chromosomal number is reduced to half. HALF reduced to half. So thereby, so here in meiosis 1, chromosomal number is reduced to half. That particular process where it is a process taking place means in Anaphase 1. In Anaphase 1, here bipolar spindle fibers are there, no? Here 2 spindles and here also 2 spindles. So what is happening now? Once you come forward, here bipolar spindle fibers or simply spindle fibers are there, no? Will come and attach at the kinetochore regions. So here and here, there. So now due to the activity of spindle fibers, what is happening? So here in Anaphase 1, the complete chromosome means without any splitting and all. Complete chromosomes start to move towards the opposite directions or opposite poles. That's why even though we took 4 number of chromosomes completely, rendu separate. Okay, so male 2 chromosomes and female 2 chromosomes separate. So that's why here in these particular 2 cells, no, we are observing the here 2 chromosomes and here also 2 chromosomes. Point purida. But my dear students, so once you come here in Anaphase 2, what is happening? Here you see there Anaphase 2, that particular information. Now let us see. It begins with the simultaneous splitting of the centromere. We have seen simultaneous splitting. So simultaneous splitting. So splitting of the centromere of the chromosomes. So here in this particular case, what are the chromosomes here we took? No. So here you see there, here telophase 2 nalli 2 chromosomes, here 2, here you 2 we took, no. Ipu later in a telophase 1 apron again prophase 2, metaphase 2 start on. Again here due to the lack of space, no, here directly I have given Anaphase 2. So thereby why I have given directly Anaphase 2, no? So I thought you are having the characteristics of prophase 2 and metaphase 2. So what are those? Prophase 2 nalli definitely thread like chromatin material condensed and converted into chromosomes. That one we have seen, no, here. And metaphase 2 here we have seen. So what is the information here? There we have seen. So what are the information? So here what is the information here we have taken, no? In metaphase 2 same information. So the chromosomes will be settled or comes onto the equatorial plate. So thereby in Anaphase 2 the chromosomes are going to be splitted. So that is the point here we have learned. Simultaneously splitting of the centromere of the chromosome, allowing them to move toward opposite poles of the cell by shortening of the microtubules attached to the kinetochore. So due to the activity of bipolar spindle fibers or due to the activity of microtubules which are attached at the kinetochore, so what is the chromosome or what are the chromosomes are there? Definitely splitted. So that's why here we drawn like this once you see. So here 2 chromosomes here we took, no. So these 2 chromosomes eppadi split ayichipare? One chromosome half avana, inge one half, inge one half vandachi. And here this particular chromosome, adi chromosome center split avana, male one half, kile one half. So likewise here also complete chromosome split avana, male one half, kile one half. Here also same. So that's why here in Anaphase 2 we have drawn this particular diagram like this. Next one, in telophase 2 what is happening? So meiosis 2 or meiosis ends with telophase 2. Meiosis ends with telophase 2 in which the 2 groups of the chromosomes once again get enclosed by a nuclear envelope. Simple one illa enna telophase 2 nalli what is happening, no, nuclear envelope or the nuclear membrane reappears. Next one reappears and nucleolus also reappears. Apro cytokinesis process. Cytokinesis process is nothing but division of cytoplasm only, no. Even a division of cytoplasm process, everything takes place and follows resulting in the formation of which one? Tetrad of cells, that is 4 haploid cells. So that's why here you see the tetra means 4 only, no, 1 2 3 4. So 4 cells of formation or 4 haploid cells of formation here we are observing in a telophase 2. So thereby once you see here totally 2 in number is equal to 4 we have taken. So here meiocyte why I have written, so where the cell undergo the meiotic division, no, that particular cell we will be considering as meiocyte. Or simply you listen carefully, the cell which undergo meiotic division is referred as meiocyte. That particular meiocyte condition also must be diploid in nature. So 100% in introduction we have learned, no, meiotic division 100% takes place only in diploid condition. So here 2n is equal to 4 here initially we took, but by completion of telophase 1 here n is equal to 2, here n is equal to 2Ig. Next one here, this particular cell, once you see this particular cell is the parent of these two cells. Once you see again, this one particular cell, this one cell, from this particular cell these two cells are there. These two cells are came now. Now for the formation of these two cells, this is a parent. So now here 2 chromosomes we are observing, and this particular cell, if you see there also 2 chromosomes are there now. But here like daughter chromosomes we will be considering, but and here 2 chromosomes, here also 2 chromosomes. So thereby, so what are the offsprings which are come from the parent, no? So parentally n is equal to 2, even offsprings also n is equal to 2, n is equal to 2. So thereby here and here the chromosomal number is equal or same. That's why meiosis 2 is equals to mitosis. So meiosis 2 diagrams, no, these 3 I have drawn for our convenience to learn. Okay, ma. So I hope you understood this one. Suppose if you are unable to understand, no worry. So in presence of you, this entire explanation, if you need to learn or if you would like to learn, suppose if you are unable to understand, I'll explain by one more time. Thank you.